Miss Nandini, am I audible?
Yes, sir, you're audible.
I think 200 plus participants have joined. The rest of the join will later on. Now you please proceed to introductory remarks.
Okay, sir.
Hmm.
Hello, everyone. A very warm and lovely good evening to one and all. I welcome you all on 30 days international training, come winter school, on the topic Navigating Agriculture Transformations for the Upliftment of Rural Excellence, that is Nature 2024. Today is 18th day of our training organized by AgriMeet Foundation Bharat in collaboration with CIMMYT Mexico, Southern Federal University of Russia, ICADA Lebanon, World Agroforestry Center for International Forest Research Kenya, ICRISAT Hyderabad, Manage Hyderabad, SKL TSHU Telangana, MGUVV Durg, HAU Hisar, IGKV Raipur, IERI New Delhi, MHU Karnal, NDRI Karnal, ICAR IERI New Delhi, ICAR DCR Puttur Karnataka, Stanford University, and University of California. It is an absolute honor to welcome you all for today's training session, where we are privileged to have our distinguished speaker with us today, that is Dr. Sean Mayers, the Global Research Program Director for Accelerated Crop Improvement at ICRISAT. Dr. Mayes is a pioneer in the field of crop genetics with profound expertise in genetic diversity and germplasm characterization across a wide array of crops ranging from wheat and oil palm to underutilized crops like bambara, groundnut, winged bean, and quinoa. His groundbreaking work in marker assisted selection and breeding approaches has played a crucial role in enhancing the resilience and sustainability of food crops. Before joining ICRISAT in 2022, Dr. Mays built a distinguished academic and professional career. He served as an associate professor of crop genetics at the University of Nottingham, where he contributed significantly to advancing crop genetics. Furthermore, during his tenure as theme director for crop genetics at the Crops for Future Research Center in Malaysia, he championed the development and adoption of underutilized crops, paving the way for agriculture diversification globally. Dr. May's remarkable journey in research spanned decades, including his leadership roles at University of Cambridge and his contributions to genetic improvement at Plant Breeding International Cambridge. His dedication to advancing crop improvement and addressing the challenges of global food security is truly inspirational. We are incredibly fortunate to learn from Dr. May's wealth of knowledge and vision for resilient, sustainable agriculture. His focus on leveraging genetic diversity to adapt to changing climates and ensuring food security aligns perfectly with the goals of our training session today. The title for today's training is The Potential for Minor Resilient Crops for Sustainable Agriculture. Dr. Mees, on behalf of everyone present here, we extend a warm welcome and look forward to your insights, which will undoubtedly inspire and guide us in our collective mission to shape a agriculture future. Over to you. The session is yours, sir.
Thanks, Nandini, and thanks to the organisation for inviting me to give a talk. Let me share my screen.
Good evening, sir.
Okay. Can everyone see that?
Yes, it's visible.
Okay, great. Okay, so what I'm going to do is I wanted to just say a little bit about where we are in terms of diversification, and particularly in terms of different crops that can be used, and just to give a few examples. So I'm, as Nandini said, I'm relatively new in ICRISAT. I've been here 2 years as Global Program Director, looking after accelerated crop improvement, which is from the gene bank work all the way through the breeding and on to the seed system side of things. So what I'll do as one of the examples, I'll actually give a little bit of a background in terms of what we're doing in ICRISAT and how we're trying to link through the different disciplines to make sure that they all align in terms of breeding. But I'm going to start really just with a little bit of context. So, I mean, you'll already know many of these things, but in terms of the challenges facing global agriculture, a little bit about the University of Nottingham and Crops for the Future, then ICRISAT, and then finally I'm going to talk a little bit about Vision for Adapted Crops and Soils, which is generally known as VAX, before trying to come to a few conclusions about how minor crops, how diversification potentially is one of the ways in which we can get more resilient agriculture. So obviously we're facing a range of problems, a range of challenges at the moment, not least the increasing population, but also significant urbanization, particularly in Africa. There's climate volatility, and at the moment, I think we're heading for about 2.7 degrees temperature increase. The volatility is a different issue and has been seen around the world in the last couple of years, maybe one of the bigger challenges. Also, there's a shifting in patterns of rain, which is associated with climate change, but also in many areas, there is limited rain anyway. So the drylands is really where ICRISAT focuses, and that covers parts of 55 countries. Part of the background in terms of some of the problems faced really goes back to the Green Revolution, which in itself was extremely good and obviously made an incredible difference in terms of human survival and availability of food, and particularly in India in terms of going from a consumer to an exporter very rapidly. But there have been consequences which relate more to when it's being used where it's not really suitable, and particularly deterioration of soil quality is a major concern at the moment. There's also increasing or certainly high levels of food insecurity and hunger. So this is from the FAO stats book in 2020. There are some more recent figures, but the trend is pretty much the same. So in somewhere like Africa, a majority of the people there are at least moderately food insecure. So they're not completely confident that they're going to have food for the lean season. But even elsewhere in many countries, and that also to some extent includes developed countries like Northern America and Europe, there's still a degree of food insecurity which needs to be tackled. A flip side of some of this, and certainly in terms of very rapid development, is malnutrition, and particularly the increase of obesity. And this relates primarily to our food systems. But obviously our food systems are built up from the crops that we grow. And certainly increase in obesity and certainly overweight individuals is very, very rapid in many countries, including many of the poorer countries. So in some places in Africa, you get obesity at the same time as you get malnutrition and undernutrition. And these are all aspects that fundamentally depend on food systems, but also clearly the crops that you grow make a difference. And really the last example is the one that's probably the most important one, is if you look at malnutrition, and particularly you look at malnutrition in terms of children. So children under 5 who are not growing well, which can be stunted, wasted, or even overweight. And clearly in India and Africa, this is a significant issue. And clearly it relates to food and feeding. And I think this is one of the ones that really should be the focus of what we look at, because ultimately those effects are lifelong. It's not simply that people will completely recover. I mean, people will recover to some extent, but certainly stunting or wasting will leave permanent effects. So it's imperative that we can start challenging these effects, and we need to challenge them through having a stable food system, but also a nutritious food system. So the first example I'm going to give is the University of Nottingham, where we have a campus in Malaysia, but we've also been working for about 30 or 40 years now on tropical minor crops. And the particular one we've been working on is called Bambara groundnut, and I'll say a bit more about that in a minute. We are also working on things like leafy amaranths, winged bean, proso millet, foxtail millet, and to some extent moth bean and quinoa. And all of these are really done as networks of different actors. So we always work with other organizations, and particularly organizations within country. And much of what we do is we look at collecting germplasm and then really screening that. And a lot of the focus is around identifying minor crops that have crop resilience. In something like Bambara groundnut, that is very much in terms of drought tolerance. But obviously leafy greens are important in terms of nutrition, and amaranth is also quite good in terms of drought tolerance. So there are a range of characteristics which potentially allow agriculture to work in places which don't have the high inputs, which potentially don't have access to some of the inputs that if you like, developed world farmers would expect. I'll just outline the second. So ICRISAT is where I'm working at the moment, and it has a 52-year history of working on 3 cereals, so sorghum, pearl millet, and finger millet, 3 legumes, chickpea, pigeon pea, groundnut, and we also work on many of the minor millets, which are still around, so certainly in India, still used, but they're used at relatively low levels compared to how they were used before the Green Revolution. And most of these have some degree of stress tolerance, certainly abiotic. The final example, just to give you the comparisons before going into detail, is really in terms of the vision for adapted crops and soils. So this is it's actually a US State Department initiative, but the idea was very much to identify those resilient crops, which are still grown at reasonable level, particularly in Africa to begin with, and to look at what we can do to actually bring those forward as alternatives. And one of the nice things about this project is it's very much focused on nutrition, and it's stated very explicitly nutrition is an important part of this. So for one of the examples, I'm going to talk about some work at Nottingham and elsewhere, which is on Bambara groundnut, really just as an example of an underutilized crop. So it's one of the Vigna. So there are lots of common related crops across the world. The closest one is probably cowpea. So its strengths are things like drought tolerance. It grows in very poor environment soils. It's nitrogen fixing as a legume. It's relatively fast, 4 to 5 months, and it's probably the 3rd most important nutrient legume in sub-Saharan Africa. And it's grown basically by millions of farmers. In many cases than in the commercial side. So we've done quite a lot of work looking at germplasm collections, looking at the variability that exists, and clearly there's a lot of variability in this crop. And part of the point of doing that is to try and identify where we might identify more important traits or more useful traits. And in terms of distribution, it's distributed quite well across West and Central Africa, and also East and Southern Africa. There's a little bit in Indonesia, a little bit in Thailand, and a little bit in Malaysia. But it is basically an African crop, and it's a native African crop that's been there for a long time. So one of the first things that we really wanted to do was to identify some of the variability and to generate controlled material, controlled cross material. And in practice, this is not so easy with this crop because the flowers are extremely small and at soil level. However, it can be done, and from there you can start to look at the variability that's present between both domesticated but also wild material. And some of the wild material is potentially quite useful in terms of introducing new traits in the future. And what we did in practice was work with a number of people, a number of groups around the world, and we identified 12 individual lines which represented a good selection of germplasm. And those 12, we worked to develop crosses and then look at the segregation for all of the interesting traits to begin to understand how we would develop markers or how we develop markers to do the selection in breeding. So, as I said, crossing is not simple, but it's certainly possible. So making a range of crosses, it became possible to generate F1s. By being very kind to those F1s, we could get up to sort of about 800 seed, which is really very high numbers in terms of an individual Bambara plant. And we also had markers, so we developed SSR markers and were able to use those, first of all, to show that it was a hybrid, because it's not really that obvious. And I think that that's one of the things that has become pretty much endemic in terms of breeding programs now, is to confirm the hybrid as soon as you can. And then worked through a number of generations. We have tropical glasshouses in Nottingham, and we've used those really to look at a range of traits and to look at water use, to look at the effects of temperature, to look at the effects of different soil composition and nutrient content. And some of the material from those early crosses and experiments are now currently in selection in the field at late generation. So working with the University of KwaZulu-Natal in South Africa and University in Nigeria, Nsukka, and also the Kirkhouse Trust, we're basically looking at trying to develop varieties. So at the moment, there are no varieties which have been developed by crossing. But this is something which we're making progress on. Colleagues in Kirkhouse Trust are also looking at whether you can use chemical emasculation as a way to improve crossing rates. So it's doable. It just hasn't been done previously because it's difficult. And we're hoping that that leads to uniform but good material, and that good material becomes available for farmers. Because when you want to give material to farmers, most countries have regulations which basically say it has to go through assessment. And that's quite difficult if there's a crop that hasn't really been assessed before. One of the interesting problems we came across, and in many cases underutilized crops do have problems. They do have either a lack of some domestication, or they do have a slight quirk in terms of how they behave. So Bambara groundnut's an odd one in that it has an effect of photoperiod. And because we have triplicated glasshouses in Nottingham, and these are tropical glasshouses with soil pits, we were actually able to impose different photoperiods. And one of the effects of those photoperiods, if it's wrong in Bambara groundnut, is not that it delays flowering, although it can do. It actually prevents pod set. So you get almost certainly get germination, but you don't get pod development if the photoperiod's wrong. And originally this was thought to be relatively simple, but limiting what could be done with it. So that in a sense, if you want to grow it, you can grow it at the equator, but you could only grow it outside of the equator if effectively you had about 12 hours of day length at pod set. So it limited where it could be grown. So we did some quite detailed experiments looking at the behavior of the same material, but under different photoperiods. And this diagram just gives you an example. So the top three figures are one genotype called ANGPA4 under three different photoperiods. So 12 hours is the normal photoperiod or recommended photoperiod. 14 hours, so for ANGPA4 you get basically nothing. 16 hours, you get nothing. However, if you look at different genotypes, so Uniswara red, which is the middle one, you can see the pods, and the pods themselves are red. At 12 hours, you get reasonable pod yield. At 14 hours, you still get some pod yield, but at 16 hours, you don't get pod yield. And then DIP-C, which is an interesting one, you get reasonable pod yield at 12, you get some at 14. However, if you use 16, you actually end up with greater yields, although it does delay the maturity time at some point. So it was clear that the photoperiod sensitivity in terms of pod filling, which obviously is a fairly critical trait for any sort of crop, was a little bit more complex than had been understood. And that in fact, we could potentially target higher yields through using the photoperiod. At the same time, we think we've identified, and we're still testing, some photoperiod neutral material. And hopefully that potentially becomes a resource we can use in future breeding programs. In terms of stress, I mean, we've done a lot of work looking at different aspects of this. And certainly one of the key things in terms of Bambara groundnut is it has a very extensive root system. It's very deep and it develops very quickly. And that obviously, as you can understand, is key in terms of actually accessing water as the water profile drops during the dry season. But there are other variabilities there, other differences. So certainly some have different leaf to root ratio, and some actually have very small leaves and small roots. And different genotypes work better in different environments. So in Indonesia, where actually the bigger danger is actually you get, because it's grown during what is usually the dry season after rice, the biggest danger is that you actually get flash flooding, in which case legumes really don't like flooding very much. So having a different root morphology may actually be quite important, and it may not be the depth that's needed in that case. And that's one of the nice things with working with accessions of underutilized crops, is they've adapted to the environment they were collected in. And so you can find all sorts of different traits with them. We've also looked at water use efficiency and photosynthesis, and there's a lot of variation there as well amongst the different crosses. So we have material, we have markers that can be used to bring those together into single individuals, depending on the environment, depending on the reasons and the timing that the plant is being planted. Even with the genetics in place, if you like, one of the questions becomes, how do you move that through the systems that exist? And during the time, and it's been nearly 20 years now, that there's been quite intensive work in this area, we've worked with a wide range of partners, both from Africa and from Southeast Asia in particular. And those partners have provided different environments, but also different perspectives and a better understanding why people grow it, what people are looking for, and particularly farmers are looking for. And I think that understanding is really quite critical in terms of going into breeding, understanding what the farmers, what the processors, what people who might want to use it or eat it actually value in it. Because there's always a danger that you produce something, and while it may be high yielding and very disease resistant, if the farmers or the consumers don't want to eat it, they won't eat it, so it won't get adopted. So it's important to understand the social context as well as the economic context. It also has to make economic sense for farmers. And one way that can be helped, and this is a relatively new and small company that we work with at Crops of the Future, is to look at different products that could come from it. So Bambara groundnut here has been made into plant milk. It's been used as an ingredient in noodles to increase the protein content and also help flavor. And this small company is doing quite well, and it's one way that you could begin to stimulate demand, because again, I mean, there may be wonderful minor crops out there that have wonderful properties. If no one knows about them and no company is willing to work with them in terms of food products, then they really will go nowhere. So this is one example. I mean, there are other ways in which Bambara groundnut is used. I mean, it's primarily used as a household food security crop. Just a few of the people involved in that work, so both from Crops of the Future and University of Nottingham in particular. And that work is ongoing through the VAC system, which I'll explain. But again, the objective is really to try to get a good, efficient, marker-assisted breeding program running so that we can start producing material that farmers really want. But we're also using the fact that it is drought resistant and that it is relatively high nutrition as well. That's, if you like, part of the package. It has to also have other characters, because we're not trying to reproduce something that already exists, which gives high yield but lots of carbohydrate, because it's not what the food system needs or farmers need to some extent. I'll say a bit about ICRISAT. So we're a non-profit, apolitical international agricultural research institute, and we've set up in 1972. And effectively, we focus around 6 particular crops, 3 cereals, and 3 legumes. And really our vision is a prosperous, food secure and resilient dryland tropics. And really the mission is to reduce poverty, hunger, malnutrition, and also the environmental degradation which is occurring with some forms of farming. The way that we try to work is slightly different. So we have 3 programs. I mean, I look after accelerated crop improvement, and I'll say in a little bit more detail what's involved in that. But we also work with a program on enabling systems transformation and a program on resilient farms and food systems. So the resilient farms and food systems is essentially management. So it's agronomy, it's management, it's landscape level intervention, and things like digital agriculture and geospatial. And there the idea is really to optimize the available resources. And in many cases, those resources in the drylands really boils down to water. Enabling systems change not only tries to make new products from the crops and understand their nutritional value, but also tries to understand why people grow particular crops, what makes people make decisions in terms of which crops as a farmer they might grow. And also linking farm output to processors as a way of trying to increase the amount of money that goes back to farmers. And we also have quite a focus in terms of the position of women within farming. In many cases, women are responsible for household food and household farming. And also clearly in terms of empowerment of youth and the benefits to children in terms of having options within agriculture, and how do we change agriculture so that people actually want to be involved. So I'll talk about accelerated crop improvement just briefly. So we have a gene bank. We do genetic engineering, GMOs and gene editing, quite a lot of work on genomics and pre-breeding, crop physiology, crop protection, seed health, and obviously the breeding itself. We have 6 breeding pipelines. And then how do we take those varieties and turn them into large amounts of seed available to farmers? And the key thing really with this is the aim is that each crop breeding team works as a team across all of the disciplines. And that's because without taking a fairly holistic view of development of a cultivar, it becomes really quite difficult not to end up with things that have weaknesses. So the idea is very much that everybody works together to really brainstorm what's needed and where the problems could be before the breeding actually begins. In terms of gene bank, our gene bank in Hyderabad contains 130,000 accessions of 11 crops. We also have smaller gene banks in West and East Africa, which contain subsets and also help with local partners storing material. Over the last 52 years, 1.6 million seed samples have been distributed to 150 countries. And regularly, we distribute for free samples upon request. And they can go to breeders, they could go to researchers, they could even go to people who are just interested. And the aim is really that we provide the best characterization data that we have. For much of the material that we have where we've developed core collections, we also have marker data associated. And that marker data is available. In terms of gene editing and GMOs, obviously GMOs is an area that it's a little bit unclear where everything stands at the moment around the world. Even in India, I mean, the system is there, but in practice, not very much is moving through it. However, India has been quite active in terms of adopting gene editing rules. So really, we're in the position now where it's possible, once an event has occurred, and once it's possible to show that an individual plant has no DNA from a foreign source for that material to be field planted. And that really is an important step forward. And as we understand this technology, but also technology using alternative but similar approaches, because obviously Cas9 is something that's under patent, I think we begin to work out how we can make very precise changes, particularly for domestication, but also for metabolic pathways. And here we just have an example of a gene edit within a specific target zone. One of the things that is really important is understanding something about when we use selection, when we select for things, what are the effects? What are they doing? If we get high yield, why are we getting high yield? Because that potentially opens up a number of alternative routes in terms of producing yield or producing better nutrition. So really being able to analyze, and to some extent analyze at high scale, is one of the key things, and probably now is the most limiting thing, because a lot of the genetics we can do, certainly in terms of markers and genomes, but the traits are really important because they obviously interact with each other. So we have a lysimeter, which allows us to measure water use efficiency. And we also have a LisiScan, which basically allows us to make images of up to 5,000 lines. And this is just an example of how some of this high throughput technology can work. On the left, we have an example of by conventional field trialing of identifying a hotspot in chickpea, QTL hotspot for drought tolerance. The same results came out of actually using the LisiScan, but it took 5 weeks rather than 4 or 5 years. And this is the sort of area where if we can get the phenotyping right, we can move very rapidly, at least in terms of producing material that can be tested. As we do that, the other thing that we need is we need low-cost ways of identifying values within materials. So we do a lot of NIRS-based assessment of different components such as protein, starch, and fat. We also use XRF for minerals, so iron and zinc in particular, but calcium as well now. And we can physically identify what's happening because we've got CT imaging, which is actually now used directly to look at shine percentage in groundnut. And these sorts of approaches which speed up phenotyping are really quite important and are really where we need the breakthroughs, if you like, at the moment. It's the ability to get, it doesn't have to be a perfect phenotyping system, but it's the ability to rapidly get approximate values. And we can always use something a lot more specific, but slower once we know where we are with a set. Genomics, has been a lot of work on chickpea, there's been a lot of work on pigeon pea, and we now have quite extensive resequencing panels which are available to anybody who's interested. And we also have tools like genotyping tools like mid-density panels, which can be used for a whole range of different approaches, either generating markers in material that doesn't have marker data, or an understanding of genetic diversity, or a whole range of other things. We also have QC arrays, which are quite small SNP arrays, but they'll allow you to tell whether your attempted hybrid is a hybrid. And those get fitted into the process of breeding. And that's alongside specific trait-based markers or trait-focused markers. Everything we do has to go through disease resistance testing, and obviously the more and more we introduce large-scale agriculture based on monoculture, the more problems we are likely to have with disease responding to that and overcoming those resistances. And it's important that we routinely screen for the critical ones, as well as starting to look for new sources of resistance. We have a small climate change facility, which allows us to look at what happens at different CO2 levels or different temperatures. And there are a number of polytons which actually allow us to have a gradient of temperature alongside a gradient of CO2. So we can start to understand not only how does the plant respond, to high CO2 and a higher temperature, but also how do the pests respond? How do the pod borers and other things respond? How does it change their life cycle, and how is that likely to have an added impact? And then the final recent development, or relatively recent development, has been speed breeding for different crops. So we do speed breeding work on a range of crops, and really what it allows us to do is by manipulating the light intensity and the duration and the wavelengths, we can get individuals to actually flower a lot earlier than they would do normally. And this works well for a number of species, particularly chickpea. Chickpea, we can get 6 generations a year. So in theory, we could go from an F1 to essentially inbred lines in a single year. We won't have done any selection. There's all sorts of other issues potentially with that. But the more markers come in, the more we can start to use those as surrogates for the phenotyping to begin with, and then we confirm phenotyping at the end. And the crop breeding, I mean, ICRISAT has been releasing material as cultivars for a very long time in a really broad range of countries. And it's been one of the things it has focused on, and I think is important to consider, is nutrition, and particularly biofortification. And the reason for doing that is ultimately nutrition is clearly important, particularly if farmers are eating part of their own produce. And particularly it can address some of these questions of stunting if they're caused by micronutrient deficiencies. But also, it's a positive thing to have in there anyway. At the moment, there's no real market for nutrition or nutrition value added. But I think the more people understand the value, the more it becomes possible that farmers could get a premium for growing certain things. And I think that's an important way for it to go forward. The final step of all of that is really breeder seed production. So once you go through varietal registration, you have to find a way to provide seed to farmers. And many varieties really get nowhere, partly because that system isn't in place, or partly because the farmers aren't aware of them, so the demand isn't there. And this is just one example. This is actually from Malawi, where we produce a significant amount of seed based upon orders from seed companies or from government. And the idea is to be able to provide the farmers with the best possible varieties that suit the environment they work in. And seed systems is one of the bottlenecks at the moment in terms of getting genetics from the breeding through to farmers. Very briefly, try and illustrate how some of this fits together. So this is an example from Bihar, where we're co-developing a Centre of Excellence for Millets. And it has a number of work packages, one of which is really to look at material available, both in gene banks, but also within Bihar, collect it and conserve it. Work package 2 is very much about breeding and development. Work package 3 is about how do you develop those seed systems to make sure that the material is available. WP4 is all about management, mechanization. How do you reduce the amount of work involved? How do you get mechanization at a sensible scale available so farmers don't have to deal with all of these things by hand? And then 5 is really looking at how do you set up processing, both small processing units, but also potentially larger units to develop products. And in most cases, we focus those around communities and cooperatives. And then finally, we have a section on gender responsive capacity building, which is make sure that what we're doing is not negatively affecting women, because often a lot of the work is with women. So it's important that when we design something or we breed something, the aim is to reduce workload rather than accidentally increase it. And that also feeds into the community sides of things in terms of employment and production. So how can you take these things forward? Just a quick picture before from last year of the farm under development. So this is in near Gaya in Bihar. And one of the things I want to point out here, and I think this is where we are moving, obviously in India, there's a lot of rotation and the whole agriculture is focused around multiple crops in many cases. One of the things that we're trying to focus on, and we're sort of illustrating in Gaia and elsewhere, is the idea that you can alter the site to capture as much water. And then you are based upon what water you have, you can actually work out what can be grown. And you can work out the water budget for all of these crops or all of these rotations. I think the key thing here is the change here, and it's slowly coming through in the way different organizations are working, is to think of it in terms of a system rather than a single crop. So it may be that you breed millets, but that millet has to be compatible with the chickpea that comes afterwards, or the lentils, or whichever crop. And really, you're looking at what's the production per unit of land, maybe over a year or two, rather than what's the production and value of a single crop. And I think we'll see much, much more of that happening in the future as researchers begin to tackle some of these slightly more tricky things of integrating. Very briefly on Feed for the Future. So, as I said, this is a US State Department and the Foreign Commonwealth that have also added money to this. And there are basically 7 crops selected. So Bambara groundnut, finger millet, sesame, pigeon pea, okra, amaranth, taro. And each of those crops will be led by 1 or 2 CG centers alongside lots of national partners. And really what the aim of it is to ask 4 fairly basic questions. So where should we plant? So in terms of what makes sense and what's sustainable, what to plant for productivity and nutrition, what management systems are needed to make that efficient and productive, but also sustainable, and how do we apply that to a local condition? And I think this context of localism, of trying to not change the world to fit our crops, but change our crops to fit the world, is really a new paradigm, which I think is very much coming through. So initially, this was started by looking at a whole range of underutilized crops focused on Africa. And lots of people had preferences because they work on a particular crop or they used to eat a particular crop. So one of the things that's a little bit novel in terms of how they were selected is there was modeling done using AgMIP to try and understand how these crops would behave in 2050. So really focusing on the future and what the effects of climate change are. So how will the yield of these crops develop? And for each of those crops, a yield map was basically shown across Africa to give an idea of where those individual crops are likely to be more productive or where they're likely to really not work. And this is just a simple example from Bambara groundnut, where they've looked at genetic, nutritional, biophysical, really to say what works, what doesn't, and overall what crops should be there. And overall, something like 31 crops have been selected, and 7 so far have been funded. And these are 7. And these have been renamed opportunity crops on the basis that underutilized and neglected sounded a little bit negative. But they are. I mean, generally, they're resilient crops. They're locally adapted and locally accepted. But they have potential elsewhere as well. And I think that's one of the key things is all of this is about providing new options. And I think that's really what we need, because everything is so dependent at the moment on 3 cereals and 1 legume, if you include soybean. And we really need that diversification. So if I were to summarize at this point, I would say, I think it's fair to say business as usual, which means high intensity monoculture of a few crops, is not going to be possible in many cases in agriculture in the future. There are just too many places where it no longer works well enough. And certainly current major crops are water and input hungry, but they can be very productive. So where they work sustainably, it still makes sense to grow them. It's just that there are many areas where they don't work sustainably. Almost all of the breeding research focus over the last 50 years and has been on single crops. So each person has an interest in a particular crop or has an interest in breeding a particular crop. And it's relatively recently, I mean, this is being a little bit unfair on the systems people, but it's relatively recently that the importance of working with systems has been recognized, and particularly in working with systems which have multiple options and multiple crops. By planting crops which are adapted to local conditions and planting more than single crops, farmers do also mitigate some of the risks from climate change, particularly sudden weather events. So for a single crop like rice, if you get a peak in temperature around anthesis, that whole crop could be sterile. But if you have multiple crops or you have multiple anthesis dates, then the chances of losing everything are slimmer. And I think the value of diversity, even before you start looking at where there are synergies between different crops, the value of diversity is it spreads risks. And the more risky agriculture becomes, the more important that's going to be. Growing and processing purely carbohydrate-rich crops is leading to a significant increase in non-communicable diseases. I mean, that's true. I mean, it's a food systems driven thing, but one of the problems is that there isn't the diversity there in the first place. So dietary diversity is important, but you can't have dietary diversity without crop diversity. And hopefully one can help push the other one. And generally speaking, crops grown for thousands of years under harsh and low input conditions are better able to cope with stress conditions because they are adapted to it. They've also kept the genes that are important in adapting to it. Whereas for some of the high input crops, where we've just given it everything it wants, those genes may have been lost because they may have had yield penalties. So I think having crops which don't necessarily produce the best yield, but produce a good and nutritious yield and are more consistent and resilient is one of the key things. VAX is interesting as a topic, and it's getting quite a lot of buy-in from other people because it potentially allows us to identify a whole range of material, a whole range of crops that are nutritious, resilient, and work with lower inputs. And in terms of sustainability, particularly in non-ideal conditions, having those options will be really very important to be able to put together cropping systems which are sustainable for the future. Thank you.
Well, thank you so much, sir. It was truly a self-explanatory and session and packed with valuable information. So yes, candidates, if you are having any type of doubt on any particular topic, the chat box is open. You can ask, and I'm directly going to pass the question to sir. You can ask your question. The chat box is open, everyone. Sir,
I think lot
students
of
are praising that very
lot of greetings are
informative
coming. Nice
presentation.
explanation. Very good presentation. If you have any doubt related to the particular topic or presentation, you may ask. The chat box is open for you.
Everyone is praising.
It's not usual I'm that clear, so
Please write questions, not greetings. Please write questions.
Sir is here to answer all of your doubts and queries. So if any doubt, you can ask freely.
Thank you, Professor Ephraim sir, for joining. Dr. Sunita madam for joining.
Thank you.
Nandini, please read chat box.
Yes, sir. That what is Bambara groundnut? Can you please explain about it?
yes, it's a pulse crop. But it looks, I mean, in terms of how it behaves, it's very similar to groundnut. So it buries its pods. It looks quite similar in many ways, but it's related to cowpea, so long bean and more usual cowpea. So it's a vigna.
Okay, next question.
Okay. Please suggest any new software for diversity analysis.
There's a lot of software for diversity analysis. I mean, I think I wouldn't be able to suggest any specific one because they either work on a different type of data or they work on a different basis. But in essence, I mean, all diversity analysis software really looks at generating some sort of diversity matrix, many of which are based upon matching of SNPs or microsatellites. Or what I would say is that in our experience, I mean, we've looked at Bambara groundnut using both DART arrays and DART-seq, which are basically genotyped by sequencing, and also looking at microsatellites. What is different between those two marker types is the microsatellites evolve a lot faster. And if you do diversity analysis with microsatellites, you see much more detail. Whereas for something like GBS, we get very clear distinction between different groups, but we don't see very much in the way of detail, simply because individual SNPs mutate at a lower rate than microsatellites do.
Okay, next, Nandini.
The next question is, can you please elaborate on significance of plant milk in ensuring nutritional security?
I think there's a whole range of different plant milks, obviously, and obviously plant milk originally was as much to do with the difficulty of either keeping milk from cows or actually people being allergic to things like lactose within that. It's a big health trend. I mean, we know for some things like oat milk that some of the material, some of the carbohydrates in there should or do reduce LDL cholesterol, so they are good for you. But each crop will have its different characteristics. I mean, we also in ICRISAT we make a milk from a number of millets, but it includes things like finger millet, and finger millet is extremely high in calcium. In fact, it's six times more calcium in finger millet than in cow's milk on a weight for weight basis. So I think at the moment I'm not sure how much of it is driven by science and how much is driven by fad or preference. But certainly different plant milks have the potential to have some really quite positive effects. But then cow's milk is also very good. So, and I think to some extent we're still not, for most products that that food companies produce, in many cases they actually, I don't think we really know how absorbable a lot of a nutrient is. And sort of the bioavailability is one of the big issues I think is going to have to be tackled soon. And we're starting to look at some of that within the food products that we produce in ICRISAT, because it may have extremely high value in terms of a micronutrient, but if you can't absorb it, it doesn't make any difference. So I think that's the next big jump when we start looking at bioavailability. and maybe we start to have a standard way of describing bioavailability, then you could look at the different plant milks and you could identify those that have the minerals that you're looking for, and at least be confident that you can absorb that.
Next
The
question.
next question is, what is the cost required for establishing a speed breeding in groundnut?
Well, it's not cheap, but it depends on scale to some extent. So in ICRISAT, we have a couple of controlled environment room size test chambers where we can change the light intensity and wavelength. That's still not cheap, but it allows you to develop protocols. And then you can look for cheaper ways. I mean, if you're dealing with a single crop, you can look for cheaper ways to develop a facility, which may mean that you don't need the really expensive lights, because to get the response you want, just needs a particular extra bulb or a particular wavelength. So while I know there's a tendency to go out and spend an enormous amount of money on a really high-tech facility, I'd have to say that I think it can be a little bit of a waste of money, particularly if you're looking at a single crop. It's better to work out how do you get that crop to behave the way you want, and then work out how do you build something bigger afterwards.
Okay, sir.
Next,
How can these crops contribute to food security and manage nutrient deficiency diseases?
So I think it depends to some extent on which of the crops. So certainly pearl millet, there are school children feeding studies which can show that you reduce anemia, or you reduce or certainly reduce the effects of stunting over a period of time through having a single meal a day. We don't know what's causing that. I mean, it may simply be that you have more iron, you have more zinc. There may be other effects. I mean, the other thing that's becoming extremely clear is that there's a big interaction between what you eat and the bacteria in your gut. And different people have different bacteria, combinations or levels of bacteria. And those bacteria can process what you eat. One of the reasons that beans tend to give you wind is because the bacteria in your gut are actually processing those. But they also process precursors to things like neurotransmitters. So there's a really complex interaction there in terms of what you eat and then what your gut bacteria do and how it affects you.
Okay,
so
sir.
Sorry.
Okay, you can continue. Sorry.
No, I've lost track of what I was saying. Where were we going?
Okay,
Okay,
next
should I read the
question.
next question? Sir, what are the main differences between major staple crops and minor resilient crops in terms of resource requirements and adaptability? How the nutritional content is being preserved in both biotic and abiotic stress conditions?
Yes. So, I mean, I think obviously we know, I mean, if we look at the calories eaten on the planet, then 60% of the entire calories eaten on the planet come from essentially 3 cereals. So those are the major crops, obviously. They've been selected largely, until recently at least, largely for yield increases. And they're the ones that you put large amounts of nitrogen on, you irrigate, and you really, you try to make their lives as happy as possible, and then they'll produce good yields. The concern, I mean, there are a number of concerns, and there is some data available suggesting that if you select purely on the basis of yield, you might be starting to lose beneficial alleles that are involved in resilience or involved in actually communicating with things like the rhizosphere, because they can be negative in terms of their effects on yield. And disease resistance is a classic one where some disease resistance genes will decrease yield. So if you're selecting purely on yield, you're potentially opening out that cultivar to being less resilient. Minor crops, again, it depends how you define these, and there's lots of arguments about how you define them. I mean, I would tend to work with the idea of focusing on minor crops that have resilience traits. So all of the millets, as far as I'm aware, have developed and have been adapted to dry land or fairly dry land environments where they can produce reasonably good yields, or at least reasonable yields. So they must be adapted to some extent with dealing with dry environments or with dealing with potential stress. And I think what we don't understand, is we don't understand how the different ones vary. And I think that's again, an area where we can begin to understand the basis for example, drought tolerance. And we can look across species to see whether the same thing is feasible or the same thing exists in other species. I think it also clearly, I mean, one of the other major changes is with many of these crops, the agronomy and the management will be different because you're dealing with something that's quite different. You're not, there's a reasonably good chance that things won't respond well to massive inputs. The sort of example that's normally given, which is sort of not really a very good example, is if you put lots of nitrogen onto something that doesn't contain dwarfing genes, it will get very tall and fall over. But there will be other cases where the plant is not using the resources well, or the resources are being used in ways that don't directly benefit the grain, if that's what it produces. Again, I think everything is fairly crop specific in many ways. I mean, the struggle is trying to find ways to look for the traits that we want that could be used across crops. But when you want to apply that in a breeding program, then you're breeding within that crop, and that's just how it is. You could potentially use things like gene editing to move traits across, and that potentially is quite an important area in the future, because I think many of the minor crops are not really that domesticated. So even in some of the minor millets, they're shattering, or they're even that they don't come easily up from the hull when you try and take the grain. So there are ways in which the minor crops could be adapted more to agriculture to make it easier, to make it more sustainable, to make it longer term. A simple example would be pearl millet, where you have about 6% polyunsaturated oils. And when you grind pearl millet into flour, it goes rancid relatively quickly because the enzymes within pearl millet start breaking down those oils. If you could knock out those enzymes using, say, gene editing, then you could potentially have flour that has a much longer shelf life. And that means you don't have to grind or rebuy every couple of weeks, depending upon how you do it. But it is crop specific, and the trick is going to be finding ways of working across crops, because the more we open, if you like, the more crops we have that could be of interest, the smaller and smaller the amount of money available becomes to do that research. So we have to find ways to work across.
Okay, sir. The next question is, what is validated marker for pearl millet?
A validated marker for a particular trait, or so I mean, the sort of things we would look for. I mean, the example of rancidity is one. So if you have a population that segregates for rancidity, you can identify the rough location of the gene causing that. You might not be able to identify the individual gene. If you have DNA markers which show differences between the parents on either side of that gene, then those would effectively be a test to see whether you carry one version of the gene, which may be producing higher activity lipase, or a different version which has a slower rancidity. So really validated markers within a crop are ways in which, having identified the approximate position of the gene, you can trace that position as you breed into the next generations and know which version of that gene you've inherited.
Okay, the next question is, please suggest some climate resilient and short duration chickpea varieties.
Very good question. I'm not the chickpea breeder, but it is one of our targets. I mean, we're doing quite a lot of work in Orissa State on rice fallow, where we're really trying to plant, and it's a big project there led by the government of Orissa, trying to plant a legume after the rice and trying to use the residual water. It works well for a number of legumes. It just about works for chickpea. Chickpea is borderline because it's a little bit too long. It does work most of the kind. If you can do 1 or 2 irrigations, it's fine. You're doing it purely on residual water, it's difficult. Certainly within ICRISAT, most of our focus, or one large strand of our focus, is to get early maturity on as many crops as we can. And certainly early maturity chickpea is something we are working on. I couldn't tell you offhand exactly where that stands, but if you drop me an email after this, I can put you in touch with the chickpea breeder.
Yes.
Okay, sir. You can share your email. And the next question is, please mention most effective climate resilient strategy, system, or plan.
I think, I mean, we're still, there are a whole range of different approaches, but my feeling that in terms of where you can do it, I think optimizing the landscape to capture the best resources you can, and particularly in dry land, storing that water so that it can be used later, rather than it simply runs off or it actually causes flooding problems. That allows you then to see what resources are available, particularly in terms of water, and actually design your cropping system according to what resources are available. Now that's obviously a lot of work, and at the moment we're only just starting to put together those different systems and understand how everything fits. Certainly working with low tillage or no tillage, whether that's regenerative agriculture or it's one of the forms of conservative agriculture, is really quite important. I think that's one way in which you can reduce soil degradation, potentially put more carbon back in the soil so they become more functional. And one area we're looking at the moment, starting to look at in more detail, is the idea of, can we actually grow perennial crops? So one of my colleagues who's actually online at the moment works on perennial sorghum. And the idea is, if you can get you can use it first of all as a fodder, so it will grow as a fodder and it will regrow by itself. But potentially down the road a little bit, we could improve the quality of the grain that comes from it. So it could be something that might last 6 or 8 years before replanting. But what you'll get is an incredibly dense root system. And if you're dealing with, areas where there could be drought or there could be other issues, that well-established root system has a lot of advantages. And it may well be for some of the areas where we're doing agriculture, that's what will happen in the future. It will be perennials, simply because the variability of the climate and the periods when there aren't very many resources or there isn't very much water mean we have to move to something that's a little bit more permanent to be able to actually ensure we don't have rapid and frequent crop losses.
Okay, sir. The next question is, what are the career opportunities for young researchers starting to work with these minor crops and how to proceed?
A good question. I think there are really some quite significant opportunities developing. I won't say that they're fully there at the moment. I think the VAX is a good example. So the VAX system also includes a scholarship system. I mean, it's focused in Africa at the moment, but it will put together certainly PhD studentships which are focused on aspects of those new crops, and obviously those people will go on to become some of the main researchers in those areas. There's all sorts of things, I think, happening or starting to happen in terms of trying to understand through modeling, trying to understand how complex systems work. The thing that, if I say where I think we don't have the capacity at the moment, I mean, I would say it's probably still bioinformatics, data analysis, experimental design, certainly phenotyping is a big one. But people who understand complexity is quite rare in a sense, because the standard way of doing science is to break everything down into small bits and try to understand it. But then someone really needs to be able to take all those bits and build it back up again.
So the next question is, in terms of fighting non-communicable diseases, do you think millets have advantage relative to major cereals? Please provide details on this.
I think that depends on what the cause of non-communicable is. So, I mean, it's a whole range of different, it's effectively a syndrome of different effects. Micronutrient deficiency, we know, and while we don't know exactly why it works, and that's one of the things we're trying to find out, we know that feeding school children a millet-based meal once a day has a significant effect. So there is something that we know already. Pearl millet has a relatively high nutrient content, micronutrient content, particularly iron and zinc. And in India as well, there's a minimum level now of iron and zinc that a cultivar has to have before it can be released. So I think there's good evidence that you can make an effect, that you can have an effect with some of these crops, that you probably can't with some of the major cereals. I mean, it depends. I mean, things are changing in terms of even the major cereals because they recognize the issue. So I mean, in rice, you have about 8% protein in standard rice, which is quite low. But they are developing or have developed some rice that has up to 16% protein. So I think there's an understanding that how we've developed the major crops has led to them having a number of deficiencies, and that people are trying to address those deficiencies. Now, whether it's better to do that through crops where you need to make quite big changes, or it's better to do it through diversifying crops is a different question. But there is good evidence. Well, I mean, it's sort of, in many ways, it's self-evident that if you have a good diet, you're likely to be healthier. I don't think we really understand exactly how a lot of that works, but there's good evidence that it does.
Next question.
Okay. I think that's all. You all have he has answered all the questions. So thank you so much, sir. I would like to take a moment to express my heartfelt gratitude for this insightful session. It was truly self-explanatory and packed with valuable information. I have also learned so much that will help me both personally and professionally. Thank you so much, sir, for sharing your knowledge and guiding us so effectively. Your efforts and expertise are deeply appreciated. Once again, thank you on the behalf
And
of
I'm very
entire.
thankful, to Dr. Sinmay sir for acknowledging my request and delivering the lecture despite being in the UK. I truly appreciate your efforts and dedication, sir. And looking
Well,
forward to your support and blessings in the future academic activities of AgriMeet Foundation, sir.
okay.
And
Thank you
thank
very
you.
much, and thanks to all of the people listening. Again, I put my email there, so feel free to get in contact if you have questions.
Thank you all
Okay.
participants for joining. And I'm also thankful to Professor Ephraim sir and Sunita madam from the same organization for joining, and Sakshi Dwivedi from KBMP for joining us. All participants can left. Very lovely good night. Thank you all of you.
Good
Thank
night.
you.